Okay, so let's start with the next keynote lecture. The invited speaker from far away, abroad, overseas. It's a really, really great pleasure to introduce to you Mark Febbraio. Mark is head of Cellular and Molecular Metabolism Laboratory at the Institute of Pharmaceutical Sciences at the Monash University in Australia. He has done really a lot of seminal work connecting the obesity, type 2 diabetes epidemic with cancer risk at different levels, basic research, but also translational research, because he is also involved in translating to potential products, being a CSO of two different companies, Engine Research Laboratories in USA, and another one that he also co-founded recently, Kinomedica. So it's really a great pleasure to have you here with us today, Mark, and we really look forward to your talk, which is entitled The Role of GP130 Receptor Activation in Metabolic Disease and Cancer.
So first of all, I'd like to thank the organisers for inviting me to such a beautiful place. I'm actually surprised some of you are even here, given how nice it is. So I take that as a compliment already. And thanks, Bart, for the introduction. What I'm going to talk to you about today is some work that basically my laboratory's been doing for about 20 years. And I'm going to take you back briefly 20 years ago and give you a little bit of a tour of the history of how we found this. So as Bart said, I have a couple of companies, and these are my conflict of interest disclosures. I'm not going to talk about any of the Engine work today, but I'm going to talk about my new company, which is called Celesta Therapeutics. So this is really a good start to what we do in the laboratory. In our laboratory, it's from a review we published a few years ago. And really it summarises, if you like, the nexus of what we do in the lab. Because I'm a trained exercise physiologist, as you'll see as this progresses. And we know that exercise reduces the risk of a myriad of diseases. And we also know that hypernutrition and lack of physical activity, conversely, increases the risks of many of these diseases. And I'll talk about type 2 diabetes, cardiovascular disease, and I'll touch on liver cancer, which isn't listed here. So we think that exercise is a preventative treatment for non-contagious diseases, but not everybody can or will exercise. So the overreaching aim of our lab is to characterise the therapeutic potential of muscle-secreted proteins termed myokines for metabolic disease and cancer. And this work had its genesis well over 20 years ago, and Professor Bente Petersen from the University of Copenhagen has been my closest collaborator in this work. And basically, we independently made a retrospective observation, and that is that in sepsis, there is a precipitous increase in the cytokine interleukin-6, but this is preceded by an increase in the very pro-inflammatory cytokine TNF. When you exercise, there is also this large increase in interleukin-6, but this is not preceded by any increase in TNF. And this is a very old photograph, as you can see here, and there's Bente and my colleague Adam Steensberg at the Lake Bank Saltine and Tucker myself, and we went on a journey of trying to work out where this IL-6 is coming from during exercise and what is it doing. And you might ask, what has this got to do with atherosclerosis? But by the end of it, you'll see there is a link. And so the first experiment we did, we placed catheters in a femoral vein and femoral artery of human beings, and they had one leg which was depleted of glycogen, depleted of energy the day before. And we brought them in the lab, and then they did double-legged knee extensor exercise to exhaustion. So you can see this is a very old study. But what we showed was that the leg that lacked the glycogen, that had less energy, produced more interleukin-6 and produced it earlier. And we hypothesised that this could have been a signal being sent out to the muscle as like a danger signal that the muscle needs nutrient. We went on to show that it was the muscle cells themselves. You can see this here via in situ hybridization and also by immunohistochemistry. It was in fact the muscle cells themselves, not the endothelial cells or the immune cells, that were making the IL-6. Now this particular study, as my career's progressed, has become my fondest study. It's also the last time I did any work, so that is also something that is there. And this is Benter and I a few years ago. But this was a really important study, and I want to talk you through this. So we exercised people at 40% of maximal oxygen uptake, so very easy exercise, and they didn't make any interleukin-6. And then we exercised them at 70% on a bicycle, and this is moderate. It's pretty hard exercise. If you took it up to 80%, the subjects may fatigue, but it's pretty intense exercise. And you can see that there was about a tripling of the increase in interleukin-6 in the blood. And then what we did is we did a third experiment where we calculated, we did some calculations, and then we did another trial at 40%, but we infused recombinant human interleukin-6 to try and match the levels at 70%. And we asked the question, does this interleukin-6 that mimics muscle-secreted interleukin-6 result in an increase in glucose production by the liver? And the answer was yes, it did. So this was a signal being sent from the muscle to the liver to liberate glucose. And it really is the first example of the muscle acting as an endocrine organ. And in a News and Views written about this paper, you can see that if there was an energy crisis in the muscle, now remember back to when we depleted the leg in that first study, more IL-6 was released. So if there's an energy crisis in the muscle, interleukin-6 is released. It then signals to the liver to make glucose. Now, at the time, everybody said, see, interleukin-6 causes insulin resistance because when you liberate glucose from the liver, that is a hallmark of gluconeogenesis, which is a hallmark of type 2 diabetes. The problem with that hypothesis is that in the post-exercise period, human beings and animals are insulin sensitive, not insulin resistant. So that didn't make sense. So the next study we did was by Andrew Carey when he was a graduate student in the lab, and Greg Steinberg, a postdoc who did some work with us. And this time we infused interleukin-6 during a hyperinsulinemic euglycemic clamp. And contrary to the dogma at the time, which associated interleukin-6 as being a pro-inflammatory cytokine that causes problems with cardiovascular disease, causes problems with diabetes, the glucose infusion rate during the clamp was actually increased when we infused recombinant human interleukin-6. Now, you can't use interleukin-6 as a treatment, and if there's one slide I'd like you to pay attention to, it's this one. It's very complicated, so I want to talk you through it. So, interleukin-6 is a very complex cytokine in how it signals. So IL-6 binds to the IL-6 receptor and it causes a homodimerization of GP130 to transduce the signal. And this is known as classical signaling. But IL-6 also signals in a different way. And in work pioneered by my colleague at the University of Kiel, Tran Signaling, there is a phenomenon known as Tran Signaling. Now, what happens is the interleukin-6 receptor can be cleaved at the membrane by metalloproteinases, such as ADAM10 and ADAM17. They get cleaved, and the IL-6 then forms a complex with this cleaved soluble IL-6 receptor. And this can then signal in cells that don't express the IL-6 receptor, but express GP130. And nearly every cell in your body expresses GP130. And this is known as IL-6 Tran Signaling. And it's the very pro-inflammatory wing of IL-6. So what you want to be able to do is maintain membrane-bound signaling, because as I showed you, IL-6 has some positive effects, but inhibit Tran Signaling. Now you can do that with a drug called elamcocept, which is in a clinical trial at the moment for ulcerative colitis, but I'm not going to talk about that today. But the other members of the cytokines, such as CNTF, they can't transsignal because they require not only their cognate alpha receptor, but a LIF receptor or an oncostatin receptor. They don't result in a homodimer of GP130, and therefore these cytokines don't participate in transignaling. So Matthew Watt, when he was a postdoc in the lab, he basically showed the same thing with one of these cytokines called CNTF, as we showed with interleukin-6. It increases fat oxidation via activation of the molecule AMP kinase. And you can see that here because when we infect muscle cells with an AMP kinase dominant negative virus, you no longer get this increase in fat oxidation and you lose the positive effects of CNTF on metabolism. Now, unbeknownst to us, when we were doing these experiments, Regeneron Pharmaceuticals had a recombinant human form of CNTF called Axokine in a clinical trial for obesity. And it was a serendipitous way they got to this. CNTF is a neurotrophic factor. So they trialled this for motor neuron disease, for ALS, and it didn't work. But when they looked at the data, they showed that the patients that got the drug compared with the placebo, they lost weight. So they repurposed their drug as a weight loss drug. But it didn't work. And it didn't work because of safety. The patients developed antibodies to this drug. And remember that this drug is a variant of a human drug. So its endogenous actions are going to be impaired if you give the drug and the patient develops antibodies. And those endogenous effects are mainly taking place in neurogenesis and in the brain. So if you're going to tamper with brain function, it's not going to fly as a drug. But the lead researcher at Cornell said there was still a huge number of people out there that can benefit from a drug like this. And so we started thinking about this 15 years ago. How can we harness this information to create a drug? And if you go forward almost 15 years, we made such a drug. And the drug is called IC7FC. This is Stefan Rosjon and this is Maria Fenderson and Tamara Allen. They were the first co-lead authors on this paper, and Stefan is my really close collaborator, who's a chemist and who synthesised this drug. And I'll explain this drug. So these are the 3 sites on IL-6. This is IL-6, this grey molecule. Now, site 1, it binds the IL-6 receptor. Remember I showed you that. Site 2 binds one of the GP130s, and site 3 binds the other. So what we did was something very simple. We cut off this site 3 loop from IL-6 and we pasted on this site 3 loop from CNTF. So now we have a molecule that the sequence is almost completely IL-6. And remember, IL-6 circulates freely. Every time you ran up the stairs from the C to come up to the meeting, you will make IL-6, it will circulate. So the likelihood you're going to get antibodies to IL-6 aren't very high. Because CNTF, this molecule here, it lacks a secretory sequence peptide. It doesn't circulate. So if you give it in the blood, that's why you're likely to get antibodies. So you can see, apart from these residues here, this is a molecule that really looks like IL-6 with some differences. So IC7 is IL-6 with one GP130 binding site substituted for the LIF binding site for CNTF. And this is how it works. You can inject it. It will complex with the soluble IL-6 receptor or at the membrane, and then heterodimerize LIF and GP130 to form a complex. So the molecule behaves like IL-6, but it can't trans signal. And these are the data we showed. Now I'm going to walk you through a little bit of published data, and then I'm going to finish with unpublished data. So the first thing we showed was that when we gave animals IC7, they ate a little bit less. So we did a pair feeding experiment to see whether all of the benefits of IC7 are simply due to reduced food intake. And the answer to that is they are not. The drop in adiposity is entirely due to the food intake. But what was really quite surprising was that even though these animals ate the same amount of food, they were given less a nutrient than the ad libitum group. You can see that they defended against any drop in lean mass. So if you have a drug that can potentially lower fat mass but protect against a drop in lean mass, particularly in a disease like type 2 diabetes, which is an aging-related disease where you get sarcopenia, this is a good thing. Now, one of the reviewers said, this is almost too good to be true. When a reviewer says that, it's usually, I don't believe you. So we had to work out the mechanism for this. And this is my colleague and friend Michael Caron, and I'll come back to this. He published a paper in Nature about 2015 showing that if you activate GP130, you can activate YAP to regenerate intestinal epithelial cells. And I remember reading this paper, half of the reason why I have some success in science is luck. Because I remember reading this paper at a conference that my collaborator who's in the audience, Robin Murphy, was also at. And they presented this paper. And this is Kevin Watt and Paul Grigorovich. And what these individuals showed was that you can prevent a drop in muscle mass by activating YAP. Or the other side of this is, if you knock YAP out of muscle, you get muscle wasting. So I talked to Paul, actually I talked to Kevin at that meeting, and we set up a collaboration, and it turned out to be a fruitful one, because what we showed was that IC7 activates YAP, it activates YAP target genes, and if you then treat muscle cells with IC7 as they grow, the myofibrillar width increases about 25% with this treatment, but not in cells where YAP has been knocked out. So this could be a treatment in itself for sarcopenia. And we followed this up. And the other thing that's important, I was worried, because Bart will attest to this, that if you activate the YAP in the liver, it's not a good thing. And if anything, YAP was down in the liver, so that was a good thing. Now, in unpublished data, we've gone on to inject mice with a control or an IC7 FC vector, the same mouse getting one limb with a control, one limb with a vector. This is a vector that is inducible with doxycycline. Then we waited 35 days, did functional experiments and looked at mass, and we were able to replicate what we showed in the paper in vivo, and that is that muscle mass relative to body mass increases, but only in the limb that got the IC7 only worked in the limb where YAP was not silenced. And you can see here the change in muscle mass from control. Now, we've also gone on to show that IC7 can increase human myoblast proliferation. So we've got human iPS-derived myoblasts, treated them, and looked at BRDU, and this is preliminary, but total nuclei go up. So it looks like that IC7 has this proliferative effect in human iPS-derived cells as well. So this is all positive. And finally, in a MDX mouse, we found that IC7 treatment reduces the pathology in these mice. And we have to follow up this work. But it looks like that IC7 has some therapeutic utility for sarcopenia. And we're also now looking in models of cancer cachexia. So I want to come back to the liver now, and Bart said he wanted me to talk a little bit about liver, so I will. What we showed here, that even if these animals ate the same amount of food, they had less triacylglycerol and less diacylglycerol in their liver. They had a normalised, if you will, a fasting glucose, so back down to levels you see on a chow diet. And their glucose tolerance was profoundly improved. They had an increased fat oxidation in the liver, we think via AMPK, as AMPK activity was up. The genes involved in lipid synthesis were down, and the lipids in the liver, the livers just look so much more healthy. And this is why we think YAP is down in these animals. Now, of course, NAFLD in itself is not really that big of an issue for liver disease. It's referred to as bland steatosis, and Ben, a fellow Belgian, is in the lab now, and he is doing some great work in this area. He published this review when we were all bored of our brains in lockdown, so we wrote a lot of reviews. And you can see here that NASH really only progresses if you have inflammation, alterations to the gut microbiota, which I'll talk about, fibrosis and ER stress, which I won't talk about. But this is Michael Caron, my colleague, and this is the mouse that we use for NASH. And it's a It's called the MUP-UPA mouse and expresses high amounts of urokinase plasminogen activator, specifically in hepatocytes, and it undergoes transient ER stress. So it was actually developed by Eric Sandgren, who is in Wisconsin, simply to study liver regeneration, because the UPA gene is only transiently expressed, and after 16 weeks, the mice are normal. Now, there's several papers where we've described this mouse, and if you leave these animals on a low-fat diet, they're normal. They're slightly less body weight, but other than that, they're fine. But if you put them on a high-fat diet, they get a similar level of steatosis, but at 24 weeks, they develop all of the hallmarks of human NASH, and if you keep them on the diet for 40 weeks, they develop cancer. Now, why is that important for this talk? I'll convince you, hopefully. Now, whilst people think that it's a high-fat diet that causes this explosion we're having in NASH, the rise in NASH, if you look in the United States, coincides with an increase in the consumption of high fructose corn syrup. Now, I believe that high fructose corn syrup is banned in Europe, and it is in Australia, but it's still available in the US. And if you drive through the Midwest and you look at all the cornfields, you'll know why. So it has a lot of negativity with it. And one thing that it does is it's very toxic in the gut. It enters the apical pole of the enterocyte through GLUT5, and it really causes a lot of toxicity in the epithelium, as I'll show you. And then, after it's done its damage in the epithelium, in the enterocytes, it then is facilitated into the liver, where it can undergo de novo lipogenesis, etc. Now, in this study, Jelena Todoric showed that if you give them MUP-UPA high fructose diet, it's almost the same as giving them the carcinogen DEN. You can see there that the high fructose diet animals get a lot of aggressive cancer. And they also get really bad steatosis when you give these animals fructose. Now, why is this of interest or importance? We really looked at the integrity of the intestinal epithelium, and all of the genes which encoded tight gap junction proteins were dysregulated, and the whole microbiome was completely altered. So we wondered whether this was the primary reason that these animals were getting very sick. And the shorter answer is it was. Because when we treated these animals with a broad spectrum antibiotic, we ameliorated not only the cancer, but the fatty liver, as you can see here. So, how does this work, and what has it got to do with IC7? We think that with excess fructose consumption, you get an increase in gut permeability and an altered microbiota. You get systemic endotoxemia, which can activate TNF in the liver through toll-like TLR4, and also alter the fatty acid composition that goes to the liver. Now, this is not our work. This is work by Katie Wellen and Josh Rabinovitz in a couple of papers published in Nature and Nature Metabolism. So we think that this is important. But this was, from my perspective, the most interesting part of that study. We didn't treat the animals with IC7, but we crossed a constitutively active GP130 mouse onto the MUP-UPA and gave them fructose. And what we saw was a complete diminution of disease. So by activating GP130 only in the intestinal epithelial cells, did we see steatosis go down and basically the cancer go away. I won't delve into this. Now, one thing that we've started to do now is see if IC7 can mimic those results. And we've only done a couple of experiments, but we've done one with Bernd Schnabel, who doesn't work on NASH, but ASH. He looks at ethanol. And ethanol is a major contributor to liver disease. In fact, if you think about the patients who develop cirrhosis, they usually have a combination of a poor diet and too much alcohol and a lack of exercise. So we've done some preliminary experiments, and treating the model, the ethanol-induced model, with IC7 decreases triglycerides and decreases ALT. Now, there's one other reason other than the barrier where IC7 could be working, and we were provoked by this paper from Marcus Gonçalves, who actually got the opposite result in a way that we did. He showed that if you give very small amounts, we gave fairly high doses of fructose, and that made the liver and the gut toxic. But if you give small amounts of fructose, you increase intestinal cell survival and you increase nutrient absorption, as you can see here by this fat tolerance test. So if you give high fructose, high sugar, you get an increase in villus length and an increase in absorption. And that's really good if you're a farmer trying to make a cow fat, but it's not very good if you're an endocrinologist trying to make someone who's overeating thin, because it's exact opposite to what you want. So we asked the question, maybe IC7 does the opposite to this and acts like a pseudo fibre diet, where you get a decrease in absorption. And to do this experiment, Gideon and Michael simply used a bomb calorimeter and looked at the animal's fecal metabolism. Now, the first thing is that there was a small but non-significant, in this instance, change in food intake. So for all intents and purposes, they ate the same in this experiment, but they still lost weight. And their fecal output was the same. So it wasn't like the IC7 is giving diarrhea, like the drug Orlistat. But amazingly, the fecal caloric density was markedly increased with IC7, and the digestive efficiency was decreased. So we really think this is worth following up too, because it could be a better drug than Orlistat. Of course, these are just in mice, and we don't know what happens in humans, but it is preclinical, and we are excited. And it seems when you put the animals on a high-fat diet, the colonic length shortens, and it seems that when we treat with IC7, this comes back, but this is also very preliminary. We've also done work in non-human primates, and I won't bore you with this. This is in the paper, the Nature paper. There doesn't seem to be any sign, at least in primates, of immunogenicity, but we're examining this further. So this is Mark Donath. I think he reviewed our paper, and he wrote this nice news and views. But one thing we always worry about is what happens to the cardiovascular system. And given this is an atherosclerosis meeting, I'm going to finish with some data to make you interested, hopefully. So we've been doing work with Melina Schonke and Patrick Renson at Leiden University. And I'm really excited by these data. Now, if you want to ask me about the nuances of atherosclerosis, I don't work in this area. This is a collaboration. So I will do my best, but I will take some questions on board and maybe ask Patrick. So what Melina and Patrick did is they used the female ApoE3 Leiden CETP mice, which is, they exhibit humanized lipo, you know this. We treated them with 1 milligram per kilogram IC7 or vehicle twice per week for a period of 7 weeks. Now, the first thing that we showed was in this study, food intake wasn't different and body weight wasn't changed. So in this model, we don't see those, and it was 7 weeks, 1 injection a week. But we do see a small decrease in fat mass, as we showed. It's not significant, but I think this is a type 2 error. But the reason why the body weight doesn't change is because, again, we see this increase in lean mass and muscle mass. And this is a very robust finding. Now, in this particular study, plasma triglycerides didn't decrease, and nor did plasma HDL. But total plasma cholesterol was significantly decreased after 3 weeks and 7 weeks of treatment. So now we have a drug that's good for sarcopenia, it's good for maintaining muscle mass, has positive effects on the liver, it improves metabolism, and now it decreases cholesterol. And when lesions were looked at, the total average lesion area was decreased by 65%. And in the lesions that were present, they were milder. So this is really starting to look pretty good. So we wanted to take this a little bit further. And this time, Melina and Patrick did a similar study, but what we looked at here were Vldl production. So they did a Vldl production assay. And what you can see here in this follow-up study, triglycerides were improved after 3 weeks. Plasma cholesterol again was decreased after 3 weeks. So the in vivo Vldl production assay was to incorporate trans-labeled S-methionine into newly produced ApoB. Injections of the LPL inhibitor toloxapol was administered, and the accumulating plasma triglycerides were collected before nascent VLDL particles containing the label in the ApoB were isolated by centrifugation. So total plasma triglycerides were lower after the 90 minutes, and the labelled ApoB production was decreased by IC7 after 90 minutes. These data suggest an overall reduced production of VLDL particles rather than the production of smaller particles. Now, the VLDL, the triglycerides incorporated into VLDL were lower, but the triglyceride per ApoB particle was the same. But this was not the case for cholesterol. Cholesterol had a lowering of the VLDL, a total cholesterol, but the VLDL total cholesterol ApoB ratio was also decreased, suggesting that lipidation of ApoB is unchanged, but reduced VLDL cholesterol content indicates a primary effect on cholesterol metabolism. Now, we're following this up with a few more experiments, but we think this is pretty exciting. So to conclude this part of the talk, the robust reduction of plasma total cholesterol we previously observed after 3 and 7 weeks of treatment is preserved and now paired with a profound reduction of plasma triglycerides. Hepatic VLDL ApoB production is markedly reduced, while ApoB lipidation is unchanged, as I said, suggesting an overall reduced production of particles rather than production of smaller particles. And triglyceride content per VLDL was unchanged, while VLDL cholesterol content is reduced, suggesting a primary effect of IC7-FC on cholesterol metabolism. So it's not only a promising therapeutic for metabolism, but also cardiovascular disease. And I think this really is, I know we hate to use the term exercise mimetic, but this, remember, the genesis of this whole project was the observation that exercise increases IL-6. So we now are with the new venture. We have 3 therapeutic indications, and we are trying to make bio-better compounds. For cardiometabolic disease, as I discussed, for diseases of the gut and liver, like alcohol-induced disease and NASH, and also for sarcopenia cachexia. So these are the 3 therapeutic indications that should keep me busy till I retire. So with that, I want to thank all the people that did the work. This is my lab. They're a wonderful group of individuals, all my past and present, particularly Stefan, Michael, Benta, Patrick, Dan Drucker, who did a lot of work on the paper, my local collaborators, and thank you for your attention.
Impressive presentation.
Thank you.
Do we have questions?
Very impressive, Mark. Do we have any idea of the immune tolerance of the IC7? You just mentioned rapidly, but
yes, we do.
Eventually, in human, how would that behave?
Okay, so the answer to that question is one never knows until you go in humans. But you can try and mitigate the risk. And we've done a number of experiments where we've taken PBMCs from patients with type 2 diabetes, treated them with IC7 and with PHA as a control, and tried to look at all the immune responses, and nothing came up as a red flag. That's the first thing we did. The second thing we did and I showed the slide, but it was busy and I didn't talk about it. The study we did in the non-human primates wasn't a metabolism study. It was like a phase 1 in monkeys. And that's precisely what we did over 7 days. Now, the third thing that we're doing in making the bio-better compounds, we're using artificial intelligence programs that will find residue-to-residue linkages in the proteins and identify them as a possible immunogenetic hotspot. And then we're going to substitute amino acids that way. So we do 3 iterations of the new, and the last one is to do a deimmunization. Having said all that, we'll never know until we go into humans. But I do want to emphasize that this molecule is 90% IL-6, which freely circulates, and very few individuals ever get an immunogenic response to IL-6.
Thank you very much for your talk. From the human point of view, you told us that this molecule can be good for cancer, for hepatic cancer, but also good for atherosclerosis. But for me, what I studied is that the inflammation is good for the cancer, for some part of cancer, because it kills the cancer. But in the other way, inflammation is not good for atherosclerosis. So how this molecule can, you told us that it just doesn't seem to modulate the inflammation, but it's IL-6 still?
No, it's not IL-6, it's IC7.
Yeah,
No, it's not IL-6. So it's really important. You go back to that slide that I said, this is the most important slide, because all of the inflammatory processes of IL-6 are through Tran Signaling.
Okay.
And this molecule can't trans-signal. Okay, that's number 1. Number 2, it's not a cancer therapy.
Yeah.
It is, we think, a metabolic therapy that can block NASH that is a precursor to liver cancer. It's not a cancer therapy.
Yeah.
So that's the other thing that maybe I didn't make clear.
Okay, so so if it's from a metabolic point of view, how do have you studied in deep the gut microbiota? Because it can be linked to the gut synthesize some metabolites that can be linked to NASH and also everything.
Yeah, we're actually studying that now. In fact, we have, in our fructose model, we have identified not only LPS, but many metabolites that are produced from commensals that go up with fructose feeding in our model. We only have preliminary data with IC7 in our model, and it seems to suggest that some of those processes are positively affected by the treatment of IC7. And as I showed you, we think that because the IC7 is having an effect on decreasing intestinal absorption, this is like akin to what you will see with a vegetarian diet. And when you see those, you get positive things from the gut, like acetate being released, and the negative things by that sort of dietary intervention goes down. So I can't answer your question fully because we haven't done the experiments, but the preliminary data suggests that this is having a positive effect on the gut, not a negative effect.
More question?
Fantastic talk, Mark. Like a few years ago, Mark Donat published a paper showing that IL-6 from the muscle may favor GLP-1 production and then modulate the insulin secretion.
Yes.
Did you check if the insulin secretion was
Yes.
like altered by IC7?
Yes. So we were very aware of that paper. So I didn't have time to show the data, but in the paper, a whole 30% of that paper, IC7 is an insulin secretagogue, and it is through incretins, and that's why Dan Drucker was on the he did it. But interestingly, it acts. So in the islets, if you take out the islets and you treat them with IC7, they increase insulin, right? If you use Exendin 939 amide, they don't, right? And they also increase GIP. So one of the reviewers said, well, this is not really that interesting because if it's just going to act through incretins, we have incretins. So we then we used the Durko mice that Dan Drucker has, the double incretin receptor knockouts, and treated them with IC7, and they still had improved metabolism. So whilst it is an insulin, and one other thing, true, that I didn't mention, because it increases GIP, it also increases bone density in the animals, because we were worried about that too. So we've checked everything you said based on Donat's paper, and it does increase insulin, but it also increases glucagon, and that is also a pretty good thing because the animals don't go hypo, and the feeding, we think, is through activation of glucagon or incretins. It's complicated, Nicola, because I don't know why it still works in the double insulin incretin receptor knockouts. But if you do a GTT in those animals, there's no difference in the improvement. So it's not entirely dependent on incretins, but it activates incretins.
Okay. My second question is related to the systemic immune response when you treat in mice with your IC7. You didn't mention anything. What do you see any specific signature linked to your IC7?
All we see is so in the monkey study, the anaesthetic had a negative effect on like IL-4 and TNF, and the IC7 seemed to bring that down a little bit, that effect. In the PBMC experiments, it actually did nothing that was bad, and that's in the paper too. Like, you know what these papers are like. They take you 15 years and then all the important stuff's in the supplementary data. So you've really got to, if you want to look at that, you've really got to look at the supplementary data carefully because it's pretty much all in that. But as I said to the previous question, we're not going to know about the immune response in humans until we do the experiments.
Maybe a question. The model, the E3 Leiden data are very interesting, but usually when you lower VLDL production, you get a higher steatosis level. So have you checked with Patrick what the liver fat is doing? Yeah,
and the liver fat's not up in these animals. And I think because you're getting activation of AMPK and probably getting increased palmitate oxidation, we were worried about that too. But it doesn't seem the liver steatosis is up.
And also, you know, regarding the part with the myope mice, ER stress is important in those mice to trigger steatohepatitis and HCC.
Yes.
And you treat them with IC7, or you are breeding with GP130 transgenic mice. Did you observe a rescue of the UPR activation that was reported with? I know it's very preliminary. I understand, but you know that could explain also at the mechanistic level part of the results.
So we've looked at that with blocking SGP130, and I presented those data in Nice, but we haven't really so I have a PhD student doing that work now in the MUPS, and she's just finishing up the experiments now. So she's doing all of those analysis, but I don't have the data, I'm sorry.
Okay, I think we're coming to the end of this keynote lecture. Before we close, I would like to thank my co-chair, Béatrice Bailly, because she's not Laurent, as you may have noticed, so she kindly took over. Unfortunately, Laurent is COVID positive, couldn't make it, but I'd like to thank him also because he was very instrumental in organizing everything around
yes.
The visit.
I want to thank Laurent too.
Yes, so we hope he's doing well. And so thank you again for a great talk. And
thanks, Bart.
there we end the session.
Thank you.